Hi everyone, and welcome to our module on cell structure. Let's start by talking about the endoplasmic reticulum. This is a structure found inside of all eukaryotic cells. It consists of a folded membrane with sacs and tubules that is continuous with the nuclear membrane, and it is the site of synthesis of proteins and lipids. If you look at the bottom of the screen, this is a cartoon of the internal structures inside of a cell. This is the nucleus here in purple, and this folded structure surrounding the nucleus is the endoplasmic reticulum. The endoplasmic reticulum can be divided into a smooth portion and a rough portion. Let's talk about the rough endoplasmic reticulum first. On the rough endoplasmic reticulum, the surface is covered with ribosomes. This gives the surface a granular or rough appearance, hence the name rough endoplasmic reticulum. And this is the portion of the endoplasmic reticulum where proteins are synthesized, and that should make sense to you if you know that proteins are synthesized on ribosomes. You can divide the ribosomes inside a cell into those that are membrane-bound and those that are free. The membrane-bound ribosomes are found in the rough endoplasmic reticulum, and they produce proteins mostly for secretion from the cell, so things like protein hormones and digestive enzymes and other proteins that are going to exit the cell and serve a purpose extracellularly. The free ribosomes are found free in the cytosol, that's how they get their name, and they produce proteins that are mostly going to be used internally by the cell, such as proteins that are important for metabolism of the cell or structure of the cell. Shown on the screen is a microscopic image of the cell body of a neuron, and the purple things inside the cell are called Nissl bodies. Nissl bodies are the rough endoplasmic reticulum in neurons, and their role is to synthesize protein neurotransmitters. And you can find rough endoplasmic reticulum in many other cell types. This type of endoplasmic reticulum is especially abundant in any cell that secretes proteins. So for example, you can find lots of rough endoplasmic reticulum in the goblet cells of the intestines, as shown in this slide on the bottom left of the screen. Those cells are responsible for synthesizing and secreting mucus. You can find lots of rough endoplasmic reticulum in plasma cells, as shown on the right side of the screen, because plasma cells need to synthesize and secrete antibodies. You can also find lots of rough endoplasmic reticulum in pancreatic beta cells, which need to secrete insulin, which is a protein. Switching gears, now let's talk about the smooth endoplasmic reticulum. This is the portion of the endoplasmic reticulum without ribosomes, hence the surface is smooth instead of rough. And this portion of the endoplasmic reticulum is important for lipid and steroid synthesis. It's also important for detoxification of drugs and toxins. And you may have heard of the sarcoplasmic reticulum in muscle cells. The sarcoplasmic reticulum is the smooth endoplasmic reticulum in myocytes, and this is very important for storing calcium that will be used to generate muscle contractions. So if you understand what I said on the last slide, it should be very easy for you to understand that there's lots of smooth endoplasmic reticulum found in hepatocytes. The liver is the site of synthesis of cholesterol and lipoproteins, and therefore you can find lots of smooth endoplasmic reticulum playing a role in this process. The liver also contains many detoxification enzymes, including the cytochrome P450 family of enzymes. You can also find lots of smooth endoplasmic reticulum in organs that produce steroid hormones. For example, the adrenal glands produce cortisol, which is a steroid hormone, and the gonads produce testosterone and estradiol, and these are also steroid hormones, so therefore these organs tend to have cells with lots of smooth endoplasmic reticulum. Next, let's talk about the Golgi apparatus. Many people describe the Golgi apparatus as the post office of the cell because proteins leave the endoplasmic reticulum and go to the Golgi apparatus where they are modified and directed to their final destinations. Proteins leave the endoplasmic reticulum in structures known as vesicles, and they are transported to the Golgi apparatus. They fuse with the Golgi membrane and empty their contents into the Golgi apparatus. In the Golgi, the proteins are modified and they are sorted for transport to their next destination. If you look at this cartoon on the bottom of the screen here, structure 6 is meant to depict the Golgi apparatus sitting in the cytoplasm near the endoplasmic reticulum. You'll read in your textbooks about the cis-Golgi network and the trans-Golgi network, so let's talk about what those terms mean now. If you look at this picture on the right side of the screen, the blue structure at the top dotted with red is the rough endoplasmic reticulum. This section of the Golgi apparatus here that is closest to the rough endoplasmic reticulum is called the cis-Golgi network, and this is called the cis face of the Golgi apparatus. So vesicles come into the cis face from the rough endoplasmic reticulum. Proteins then leave in vesicles from the trans face. So this is the trans-Golgi network over here, and this is the trans face of the Golgi apparatus. And vesicles leave from here to go to the plasma membrane or to other places in the cytoplasm. And while proteins are in the Golgi apparatus, they are sorted and shipped by adding signal sequences to the proteins that help direct them where to go. So let's talk about some of those protein modifications that are known to occur in the Golgi apparatus. It's very well described that the Golgi apparatus can modify N-oligosaccharides on asparagine. We'll talk about this in a minute. The Golgi can also add O-oligosaccharides to the amino acids serine and threonine. We'll also talk about that. And then finally, the Golgi can add mannose-6-phosphate to proteins destined for lysosomes, and we'll also talk about that. And all these modifications likely serve many purposes, although this is incompletely understood. But the modifications may protect proteins from degradation, they may direct proteins to their target location, and they may allow the proteins to be recognized by receptors. So to understand how the Golgi modifies proteins, we first have to define some terms. So let's talk about what an oligosaccharide is. An oligosaccharide is a polymer, in other words, a chain made up of sugar molecules. So you may know that glucose is a sugar molecule, but so is mannose, so is galactose, and so is N-acetylglucosamine, whose structure is shown here. It's called N-acetyl because it has this group added on to a glucose molecule. So all of these are types of sugars, and if you link them together, you can form an oligosaccharide. A very long chain would be called a polysaccharide, but a short chain is called an oligosaccharide. In protein processing, oligosaccharides are attached to amino acids. If the oligosaccharides are N-linked, it means they are attached to a nitrogen in amino acid. And this often occurs in asparagine because asparagine has an extra nitrogen molecule. If you look at the bottom of the screen here, this is the structure of asparagine. It has an amine group and a carboxylic acid group, just like all amino acids, but its side chain has an extra nitrogen, and this is a handy place to attach oligosaccharides. If oligosaccharides are O-linked to proteins, this means they are attached to oxygen. And this often occurs in the amino acids serine and threonine because they have an extra O molecule. This is the structure of serine. It's got an extra oxygen atom, and this is the structure of threonine. It also has an extra oxygen atom, and these are handy places to attach oligosaccharides, so they will be O-linked to a protein. So let's start by talking about N-linked oligosaccharides. These are short polymers of sugar molecules that are synthesized and attached to proteins in the endoplasmic reticulum. As we described, they are usually added to asparagine because it has an extra N molecule. The oligosaccharides are then modified in the Golgi where they are trimmed or other sugars are added. This picture on the screen depicts how an oligosaccharide that is N-linked can be modified as it moves through the Golgi apparatus. So it's synthesized as shown on the left side of the image. The blue squares represent N-acetylglucosamine, the green dots represent mannose, and the red triangles represent glucose. So this is what the oligosaccharide looks like when it's initially added in the endoplasmic reticulum. It then moves through the Golgi apparatus going from left to right, and you can see that glucose molecules are removed, mannose sugars are modified, and so forth, so that the N-linked oligosaccharide is changed by the time it gets out of the Golgi apparatus and exits the trans-Golgi. O-linked oligosaccharides, on the other hand, are added to amino acids in the Golgi apparatus. As I told you before, O-linked sugars are usually added to serine or threonine because they have an extra O molecule. And for example, mucins are heavily O-glycosylated, so all this O-glycosylation occurs in the Golgi apparatus. The last sugar modification of proteins that we'll discuss is mannose-6-phosphate. Mannose-6-phosphate is added to proteins in the Golgi apparatus that are destined for lysosomes. These proteins are acid hydrolase enzymes that are needed to break down other structures. The mannose-6-phosphate molecule, which is shown at the bottom of the screen, is added to N-linked oligosaccharides in the Golgi apparatus. This triggers packaging in the trans-Golgi so that the enzymes go to the lysosomes. And the reason we care so much about this particular modification is because this process is abnormal or disrupted in a rare disorder called I-cell disease. I-cell disease stands for inclusion cell disease. It gets this name because of characteristic findings seen under the microscope in affected individuals. This is a rare autosomal recessive metabolic disorder. It is an example of a lysosomal storage disease called a mucolipidosis, and in fact, I also discuss I-cell disease in the biochemistry section in the video on lysosomal storage diseases. The onset of the disorder is in the first year of life. Children with this condition have growth failure, they have coarse facial features, and they also develop hypotonia and motor delay. You can think of I-cell disease as a Golgi apparatus disorder. It's caused by a failure of processing in the Golgi apparatus. In I-cell disease, mannose-6-phosphate is not found on proteins that need to go to the lysosome. The reason this occurs is because of a deficiency of an enzyme called N-acetylglucosamine 1 phosphotransferase, which is a mouthful. But this enzyme is necessary for adding the phosphate group to mannose to form mannose 6-phosphate. So when this enzyme is deficient, the mannose found on lysosomal proteins is not phosphorylated, and therefore the proteins do not go to the lysosome. The result is that the enzymes are synthesized, but they don't go to the lysosome. Instead, they are secreted outside of the cell. This means that hydrolases are missing from lysosomes, but you can actually find them in the blood and urine because they are being created by cells. They're just not going to the lysosome. As a result, lysosomes develop inclusions of undigested molecules like glycosaminoglycans and glycolipids, and this is why it's called eye cell disease. So continuing our tour of cellular structures, let's talk about endosomes. These are membrane-bound compartments in cells that are formed by endocytosis. Endocytosis is the process of invagination of the plasma membrane to surround a molecule. This is then pinched off to form an enclosed structure. There are 3 types of endocytosis. In receptor-mediated endocytosis, cells take up specific molecules, which are called ligands, that bind to receptors on the cell membrane. The receptors are often located in coated pits. I'll show you a picture of that in a minute. Pinocytosis is when a cell ingests droplets of liquid from the extracellular space. And then finally, phagocytosis is when the cell extends little arms called pseudopods from the cell membrane that encircle a particle. And this, as you may know, is very important for immune defense. Macrophages and neutrophils are called professional phagocytes. Their job is to take up pathogens via phagocytosis. And here's a cartoon depicting the 3 forms of endocytosis. In phagocytosis, little pseudopods form and surround a pathogen or particle and pull it into a specialized form of endosome called a phagosome. In pinocytosis, a little bit of the plasma membrane pinches off and surrounds some liquid. And in receptor-mediated endocytosis, there are little pits that have receptors. They will bind to a ligand and pull it in and form a coated vesicle, which has a coat protein. And we'll talk more about these coat proteins later in this video. Back to our discussion of endosomes, what they will often do is transport their contents to lysosomes. We're going to talk in detail about lysosomes in a minute, but for our purposes here, just know that they digest many materials. So the endosome will often have its membrane fuse or join together with the membrane of the lysosome. The contents will mix and they will be digested by lysosomal enzymes. In other cases, the endosome will sometimes go back to the cell membrane and release its contents in a different part of the cell membrane from where it came. Okay, so now let's talk about lysosomes in more detail. Lysosomes are famous for having a very acidic environment. pH can be as low as 4.8 inside a lysosome. And as I mentioned before, they have many different enzymes called acid hydrolases. There are 40 plus types of these enzymes that have been described. They're called acid hydrolases because they require an acidic environment like is found in the lysosome, and they break down substrates by the addition of water molecules, hence the name hydrolase. The job of these enzymes and the job of the lysosome in general is to break down cellular waste. It can also break down fats and carbohydrates and proteins, and it will generate simple compounds which are returned to the cytoplasm to be used by the cell. So as you can imagine, if a cell is deficient in one of those 40 plus lysosomal enzymes, this will lead to a lysosomal storage disease where the cell will build up a certain macromolecule that cannot be broken down. And in the biochemistry section, I have an entire video on lysosomal storage diseases. Moving on from lysosomes, let's talk about peroxisomes. These are membrane-enclosed cellular organelles that contain oxidative enzymes. And importantly, they have enzymes that can generate hydrogen peroxide or H2O2, and that's where the name peroxisome comes from. They also have the enzyme catalase, which can use the hydrogen peroxide inside the organelle to oxidize many other substances. This is especially important in liver cells where peroxisomes can detoxify many different substances. In addition, peroxisomes can also metabolize ethanol. This is an alternative minor pathway, not the major pathway for ethanol metabolism that I discuss in the biochemistry section. Peroxisomes can also perform some beta oxidation of fatty acids. In the biochemistry video on fatty acids, I talk about how mitochondria are very important for beta oxidation, but some beta oxidation also occurs in peroxisomes. Peroxisomes preferentially oxidize longer fatty acids, and the mitochondria preferentially oxidize shorter fatty acids. Next, let's talk about proteasomes, which are structures inside of cells that destroy aberrant proteins. These are proteins that have become misshaped or misfolded. Proteasomes are barrel-shaped structures that look like this cartoon on the screen here. They're a complex of multiple proteins made up of multiple protein subunits, and they use ATP to break down aberrant proteins. Proteasomes mostly destroy proteins that have been marked by a structure known as ubiquitin. Ubiquitin is a small protein found inside of cells that tags damaged proteins, and once tagged, those proteins will be taken up by the proteasome, which will break them down. And you should be aware that there's a lot of basic science literature that the proteasomes may play a role in Parkinson's disease. There is evidence of central nervous system cells having reduced ubiquitin proteasome activity, and this may lead to toxic accumulation of proteins in neurons. For the rest of this module, I'm going to talk a little bit about how structures are moved around inside of cells. And first, I want to talk about something known as the secretory pathway. This is a series of steps for secretory proteins. Begins with translation of the protein from mRNA in the cytosol. The protein then moves to the endoplasmic reticulum lumen. It then goes to the Golgi and enters a vesicle exiting the Golgi, and then via exocytosis at the plasma membrane, the protein is secreted into the extracellular space. The secretory pathway relies on something known as signal sequences to help move proteins around the cell. Signal sequences are short molecular sequences found on proteins that are undergoing synthesis. In other words, proteins going through the process of translation. And they are used to pull free ribosomes in the cytosol to the endoplasmic reticulum membrane. This is what creates the rough endoplasmic reticulum, and it leads to proteins entering the lumen of the endoplasmic reticulum. Many of these will ultimately be secreted via the secretory pathway, although some will also stay in the endoplasmic reticulum or go to other organelles. So in other words, when you have a free ribosome out in the cytosol, and it begins to produce a protein, if that protein has a little signal sequence on it, then the entire ribosome will be pulled onto the membrane of the endoplasmic reticulum, and this will direct the protein inside the lumen of the endoplasmic reticulum. These signal sequences are short peptides or proteins. They're found on the N-terminal portion of a protein. Recall that proteins are made up of amino acids like what's shown on the screen here, and amino acids have an N end and a C end. So it is this N-terminal end where you find these short peptides that are the signal sequences. And as I said before, they direct the protein ribosome complex to go to the endoplasmic reticulum. The reason signal sequences can direct proteins and ribosomes to the endoplasmic reticulum is because signal sequences are recognized by signal recognition particles, or SRPs. SRPs are ribonucleoproteins, and they are found in the cytosol. They are complex particles made up of many proteins and RNA. Basically, SRPs are always floating around in the cytosol looking for signal sequences. When they see a signal sequence attached to a protein, they recognize the sequence, and they move the protein and the ribosome to the endoplasmic reticulum from the cytosol. In the endoplasmic reticulum on the membrane, there are structures known as SRP receptors. These bind to the SRPs, and then the protein is translocated through a pore of the endoplasmic reticulum into the endoplasmic reticulum lumen. So this entire process of attaching signal sequences to proteins, having them recognized by SRPs, is what allows them to be dragged over to the endoplasmic reticulum and enter the lumen so that they can then enter various pathways, including the secretory pathway. The last topic I'll discuss for this video is the topic of coated vesicles. So many of the vesicles moving around inside the cell have a protein coat on their surface that is important for their function. This protein coat is there because the vesicle is formed from specialized portions of membranes. So vesicles are formed from the plasma membrane or the membrane of an organelle, and often they bud off from a section of the membrane with a protein on it, and this will lead to a vesicle that is coated with a protein. And different protein coats are used in different forms of traffic inside the cell. This is important for directing proteins along the secretory pathway. It's also important in transporting things from the cell surface. And there are 3 vesicle coats that have been well characterized that you should know for the Step 1 exam. The first one is clathrin, and the other 2 are COP1 and COP2. Clathrin-coated vesicles are important for transport of vesicles from the plasma membrane to the Golgi apparatus. They're also found on vesicles going to and from endosomes in the cytoplasm. And then they're very important for vesicles that are involved in receptor-mediated endocytosis. I mentioned this earlier, but this is the uptake of an extracellular component into a vesicle. And the receptors in receptor-mediated endocytosis are often found in what's known as clathrin-coated pits. This is a picture of this process shown on the screen here. So there's a clathrin-coated pit here with receptors, and when a ligand binds the receptor, the pit invaginates and forms a vesicle that is now coated with clathrin that will move through the cytoplasm. You can basically think of clathrin-coated vesicles as the vesicles that move things through the cytoplasm. They move things from the cell surface to the Golgi and from one endosome to another. And for example, you can find clathrin-coated vesicles that carry LDL that has bound the LDL receptor. You can also find clathrin-coated vesicles that carry around growth factors that have bound to growth factor receptors. The other two types of vesicles that have been described are vesicles coated with the proteins COP1 and COP2. COP1 coated vesicles have been shown to play a role in what's known as retrograde transport. This is transport of proteins backwards from the Golgi to the endoplasmic reticulum. COP2 coated vesicles, on the other hand, have been shown to play a role in anterograde transport. This is transport of proteins from the endoplasmic reticulum to the Golgi and even sometimes through the Golgi apparatus. And that concludes our module on cell structure.